Synthetic biology is a combination of science and engineering. Unlike engineers building structures with materials, biologists are working with genes. What functions are usually proteins, from antibodies, insulin to enzyme. All these proteins are translated from their code genes. Genes have various size and shape, each carrying a special ability. When different gene parts are joined together, it will give a new function to the organism. All the bricks for a building should be in proper shapes. When a gene is properly cut and modified, they are called biobricks. Biobricks are standardized, convenient, and ready to be used. Biobricks are like apps. Installing a new app to a phone will give it a new function. Biobricks are installed into microorganisms. Together they build up a smart bacteria. The International Genetically Engineered Machine Competition, iGEM, is a global institutional synthetic biology challenge for undergraduates. Participating teams design and build their own operating biological systems from biobricks. We, 2013 Hong Kong iGEM team, are one of them. The inner structure of bacteria could be quite complicated, despite its small size. Every second, the bacterium is busy producing proteins from its genes. A bacterial microcompartment, that is MCP, is a closed structure in the bacterium body, like a mini flagship. It's got a protein shell. Various forms of metabolism happens inside the shell, so the reactions could be protected against disruption. A specific type of MCP, EUT-MCP, is a regular 16-side hedra encoded by 5 genes, EUTS, M, N, LK, for different parts. This 5-gene MCP is called heterogeneous. EUTS gene alone can also encode a complete MCP shell. This is called a homogeneous MCP. These genes are expressed into proteins, and proteins group up into hexamers. Thousands of hexamers will assemble to be a flagship and be ready for the voyage. Now, the E.capsi crew is on board. Some of us work on hexamers. Of the 5 hexamers, the EUTS points and terminals out of the exterior surface, like little hooks. We will hook a tag to it, so that these MCPs can be easily recognized and separated. Other members of us work off-board inside the shell. Since they hold space, it can be used for storing. PBK1 is an enzyme that links inorganic phosphate into polymers. When we put this enzyme into our MCP, the harmful phosphates will be leaded all their way into the right place and be linked together there. Finally, our little flagships will come back full of phosphate polymers, and we have thousands and millions of that from the environment. The excessive phosphates in lakes, rivers, and oceans could be recognized and stored properly, waiting to be used again.